Hello everyone, I'm Anas Al-Ubaidi and this is lecture 19 or session 19 within chapter 24 of the Advanced Medical Science Liaison and Medical Monitor Certification course program by CCRPS. The chapter is about conceptual and theoretical monitoring, while the topic for this session is about monitoring of drug invention and drug discovery interaction trials. Introduction. The creation and discovery of new drugs, new medications represent some of the most critical processes in modern medicine. Blending scientific innovation with rigorous testing to translate early stage concepts into viable therapeutic solutions. Drug invention and drug discovery interaction trials specifically focus on these developmental stages. Key points. We'll start with the invention assessment definition. This stage evaluates the ideation, synthesis, and early characterization of a new drug compound. It is essential to confirm that new entities are both patentable and therapeutically viable. And here is a diagram summarizing the drug compound evaluation funnel from bottom to top. Evaluate safety, validate mechanism, assess patentability. Or from top to bottom, assess patentability, validate mechanism, evaluate safety. And this is called invention assessment. Focus area of invention assessment, patentable novelty and uniqueness. Assess the originality of the compound through comparative analysis with existing drugs and ensure it meets the innovation threshold for securing patents. Mechanism of action validation, preliminary assays confirm how the drug interacts with specific targets or enzymes. As for example, a novel cancer kinase inhibitor underwent comprehensive assays to validate its ability to inhibit aberrant cell signaling pathways. Early safety profiles, toxicity screenings test the compound's effects on cellular, genetic, and systemic health. The outcome here, a successful invention phase, results in the documentation of compounds that are innovative and demonstrate potential therapeutic mechanisms with minimal early risks. Discovery evaluation. After invention assessment or invention evaluation, we have discovery. Discovery evaluation definition here. This stage transitions promising compounds from the invention assessment stage into preclinical or early clinical studies. Whether experts call it early clinical or preclinical studies, both are okay. The goal here is to fine-tune candidates for safety, efficacy, and manufacturability. And here is a diagram summarizing the discovery evaluation process. From left to right, transition compounds transition from the invention assessment, lead optimization, selection of viable candidates, toxicology and metabolism studies, and then the outcome. Key measures here for the discovery evaluation: lead optimization, test candidates for favorable PD/PK, pharmacodynamics/pharmacokinetic characteristics, as here pharmacodynamics, pharmacokinetics, to select the best molecules for further development. As for example, during drug discovery for metabolic disorders, optimize the chemical properties, reduce toxic side effects during animal models. The selection of viable candidates use high throughput screenings to identify compounds with enhanced therapeutic indexes. As for example, a small molecule discovery project eliminated candidates with poor bioavailability early to safe resources. Toxicology and metabolism studies run preclinical models to establish safety margins and predict human tolerability. The outcome here: discovery evaluations ensure that compounds chosen for clinical trials demonstrate strong therapeutic relevance while meeting safety criteria. Data integrity. The definition here as for data integrity guarantees that findings from invention and discovery trials are accurate. reproducible, and transparent throughout the process. And here's a summarized diagram on ensuring data integrity in trials. We have defined data integrity from left to right, implement good laboratory practices, use digitalized systems, prepare for regulatory audits, and achieve credible trials. Strategies for maintaining integrity. Good laboratory practices or practices, GLP, standardized procedures across labs, ensuring reliability of data between locations or analysis. As for example, an antibiotic trial for microbial inhibition employed GLP, good laboratory practice, compliant automated logging systems to prevent transcription errors or bias. Digitalized systems implement EDC, electronic data capture tool, to centralize and monitor laboratory output. Regulatory audit preparation ensure all records meet required standards for inspections by FDA/EMA or other global authorities. And the outcome here, sound data management ensures trials are credible, supporting their progression through regulatory channels. Steps to implement effective monitoring. Step 1, protocol development. Define objectives and endpoints for both invention and discovery phases precisely. Include validation methods for laboratory assays and chemical processes. Step 2, real-time data collection. Utilize digital dashboards for instant feedback on lab activities and bioinformatics analysis. As for example, a neurodegenerative trial tracked real-time enzyme inhibition kinetics using live analytics, preventing delays from manual reviews. Step 3, regulatory compliance. Align protocols with a specific FDA in USA, EMA, in EU, European Union, or ICH, International Council for Harmonization guidelines for investigational drugs and early stage trials. Example, a therapeutic protein discovery trial employed pre-audits to confirm GLP, good lab practice, or practices adherence before IND, investigational new drug submission. Step 4, so we have the protocol development, real-time data collection, regulatory compliance, then risk. Step 4, risk management. Mitigate known risks such as instability in compound formulation or insufficient preclinical study designs. As for example, a vaccine discovery program identified risks with bacterial expression systems and transitioned to viral platforms, ensuring production quality. And the outcome here? Following these steps allows streamlined operations, reducing errors and improving trial timelines. Tools and techniques for monitoring. We have the laboratory assays. Utilize assays such as cytotoxicity tests, bioassays, and PCR screenings to validate mechanism of action and safety. As for example, a novel antiviral underwent plaque reduction assays confirming efficacy against multiple viral strains. Computational models use simulations to predict PD, pharmacodynamic/pharmacokinetic parameters, and drug target interactions. As for example, oncology trials employed dogging studies to prioritize receptor ligand complexes for synthesis. Digital platforms centralize data management and team collaboration through cloud-based repositories. As for example, a rare disease program employed secure cloud systems to synchronize multi-center data during gene therapy discovery. High throughput screening HTS automate compound testing to rapidly identify potential candidates. As for example, a small molecule high throughput screening HTS high throughput screening HTS led to finding a selective pain relief ion channel modulator in early stages. Predictive analytics applies scoring models to rank molecules by likelihood of success based on combinational properties like bioavailability and solubility. As for example, anti-inflammatory drug discovery relied on analytics to optimize efficacy prior to before in vivo models. And the outcome here, the application of these tools, ensures precision and efficiency in identifying therapeutic candidates. Example of successful trials: antibody discovery in immunology. Early immunogenicity tests enabled the selection of potent and safe monoclonal antibodies. CNS central nervous system small molecule development avoided blood-brain barrier exclusivity issues through high throughput screening (HTS) refinement. Pandemic antiviral research computational tools guided discovery toward broad spectrum agents. Therapies in rare genetic conditions, enzyme replacement therapies accelerated IND, investigational new drug filings due to rigorous biocompatibility monitoring. And here is a diagram summarizing the diverse strategies in research. Here in biomedical research, and here we have successful biomedical research strategies. We have on the right antibody discovery, CNS small molecule development, pandemic antiviral research therapies, and then therapies in rare genetic conditions. Next, we will summarize what we have covered through this lecture through some tables. This table is about key differences between drug invention and drug discovery interaction trials, aspect for each aspect, the comparison between both. As for definition, for drug invention interaction focuses on ideation and creation of novel drug compounds. For drug discovery interaction explores therapeutic potential of invented compounds through preclinical and early clinical studies. Other aspects like key objective, trial metrics, data collection, example, application. Table on key components of monitoring drug invention and discovery. Component focus example. Components we have: invention assessment, discovery evaluation, data integrity, risk management, regulatory compliance. Table on tools and techniques for monitoring. Tool technique. Purpose example. Tools or techniques we have: laboratory assays, computational models, high throughput screening (HTS), digital platforms, predictive analytics. Table on steps to effective monitoring for drug invention and discovery trials. We have the step focus example steps. We have protocol development, real-time data collection, regulatory adherence or alignment, risk management, and reporting and submission. Real-world table on real-world applications and outcomes. Trial scenario focus outcome. The trials or scenarios we have: antibody discovery, CNS therapy, screening, antiviral research, metabolic disease program, rare genetic disorders. Application table. Scenario. For each scenario, key principle applied, key actions, timeline, what to watch out for. Scenarios we have: discovery of a novel cancer kinase inhibitor, high throughput screening (HTS) for small molecules targeting metabolic disorders, implementation of automated data logging in antibiotic development, assessing enzyme inhibition kinetics in neurodegeneration therapies, submitting compound-specific evidence for IND approval in biologic therapy targeting autoimmunity, addressing instability in vaccine drug discovery with viral vector platforms, developing a plague assay for confirming antiviral drug efficacy, simulating drug target interactions for oncology therapy using dogging simulations, screening ion channel modulators in pain management research, assigning predictive scoring to anti-inflammatory agents based on solubility and bioavailability. Next, we will revise what we have covered through this lecture via some multiple choice questions or MCQs. Question 1: what is the primary focus of a drug invention and drug discovery interaction trials? A. Measuring patient adherence to therapies. B. Investigating the synthesis and early categorization of potential therapeutic agents. C. Monitoring the marketing campaigns of new drugs. Or D. Ensuring rapid regulatory approvals for traditional medicines. So the answer is B, as these trials explore the invention of new molecular entities or biologics and assess their therapeutic relevance through early categorization and synthesis. ensuring safety and efficacy. Question 2, which method is primarily used to identify promising drug candidates during the discovery phase? Is it manual data entry, A, B, patient interviews, or C, high throughput screening, HTS, or tumor biopsy assessments, or D? No, it's C. The answer is C. HTS, as high throughput screening. HTS enables rapid testing of thousands of compounds via automated assays, efficiently identifying viable candidates for further development. Question 3, why is data integrity critical in drug invention and drug discovery interaction trials? And actually, the data integrity is critical in any kind of trial, not only these trials. So A, to increase the cost of conducting trials, B, to ensure the reproducibility and transparency of scientific processes, C, to replace the need for regulatory compliance procedures, or D, to monitor patient satisfaction with trial protocols. So definitely here the answer is B, as to ensure the reproducibility and transparency of scientific processes. As accurate, reproducible, and transparent data collection is vital to uphold scientific credibility and facilitate regulatory submissions, ensuring successful drug development. Question 4, what is a key advantage of employing predictive analytics in drug discovery? A, reducing time spent on regulatory reviews. B, identifying compounds with the highest likelihood of clinical success. C, automating manual safety assessments. Or D, simplifying logistical trial coordination. So, key advantage of using predictive analytics to predict in drug discovery is B, as identifying compounds with the highest likelihood of clinical success. Identifying. So B would be the answer, as predictive analytics uses algorithms to rank compounds based on properties like solubility and bioavailability, streamlining the process of identifying candidates with the best potential for success in clinical trials. Question 5, what is a critical risk monitored during the invention phase of biologics? Is it A, over recruitment of patients, B, compound instability during preclinical development, or C, insufficient participant consent protocols, or D, lack of financial resources for marketing campaigns? So what is a critical risk monitored during the invention phase of biologics? Definitely it's the instability. B is the answer, compound instability during preclinical or early stage development, as biologics often face risks like instability during preclinical synthesis, making early identification and mitigation crucial to maintaining the viability of the drug candidates. And here we come to end of this lecture. Thank you very much and goodbye.